Welcome to the Deep Dive, the show where we take a stack of your sources and, well, we really dig in to find those crucial nuggets of knowledge and insight. Helps you get up to speed fast. Today, we're plunging into this fascinating, often pretty overlooked, microscopic world of hemoplasmosis. Think of them like tiny bacterial hitchhikers living right on red blood cells. And while they often fly completely under the radar, sometimes they can trigger some surprisingly serious infectious anemia. So our mission for this deep dive is really to pull apart your sources and compare how this disease shows up in two really different animal groups. You've got New World camelids, so your alpacas, llamas, and then cervids, which are, deer. It's like a tale of two completely different host pathogen systems.
And what's really interesting here, what we'll likely uncover, are the, well, surprising differences, sometimes even counterintuitive ones, in how this disease impacts domestic animals versus their wild relatives.
Wow.
And this comparison, it really shines a light on some critical implications, not just for animal health, obviously, but maybe unexpectedly for human health, too. We'll touch on the zoonotic potential later.
Okay, let's unpack that then. Before we get into the specific animals, just what exactly are hemotrophic mycoplasmas, or hemoplasmas, like at their core? What makes them different from other bacteria?
Right. So fundamentally, they're this unique group of bacteria. And the key thing defining them, they're missing a cell wall.
No cell wall. Okay.
Yeah, which is pretty unusual. Instead, they figured out how to live right on the surface of red blood cells. Clever, really. And you find them pretty much worldwide, causing infectious anemia in a whole range of mammals. Now, here's a really critical point. A lot of infections are subclinical.
Meaning the animal seems fine.
Exactly. No sign of illness. But they can become quite pathogenic, cause disease, especially if the animal is stressed or its immune system isn't working properly.
The stress factor again.
Always. It raises that question, doesn't it? Why do some infections flare up and cause serious problems while others just hang out?
They're definitely seen as emerging pathogens, and, like I hinted, some might have zoonotic potential. They could potentially jump to humans.
That hidden versus overt disease aspect is crucial. Okay, let's start with the camelids. Specifically, New World species, alpacas, llamas. Here, the sources seem to really focus on one main culprit. Candidatus mycoplasma hemolame. Let's call it.
That's right, MFO. It's this small bacterium, and here's a kicker, it's uncultivable. It parasitizes camelid red blood cells.
Uncultivable. What does that actually mean in practice for research?
Well, it's a huge hurdle. It means we can't just grow it in a petri dish in the lab like you would with, say, E. coli.
Right.
Think of it like trying to understand a wild animal you can never actually capture and observe up close in a controlled setting. It seriously limits how we test drugs, how we might develop vaccines, even understanding its basic biology. We have to rely on more complex, sort of indirect methods like molecular tools. Okay, so it latches onto the red blood cell.
Yeah.
Then what?
Then the camelid's immune system spots it, recognizes it as foreign, something that shouldn't be there, and it mounts an attack. But because milolid's stuck on the red blood cell,
the immune system ends up destroying its own cell.
Exactly. It leads to the destruction of the infected red blood cell. That's the hemolytic anemia part.
So hemolytic anemia, when we say hemolytic, what's physically happening to those blood cells? Why is that worse than just regular anemia?
That's a great question, and it is a critical difference. Hemolytic means the red blood cells are literally being ruptured. They're bursting open.
Wow.
Yeah. It's generally more severe than just having, fewer red blood cells because the body is actively destroying its own oxygen carriers. And this destruction can be rapid, quite violent, biologically speaking. It can totally overwhelm the body's ability to make replacements.
So it's a crisis.
It's a real crisis for their system, leading to a dangerous drop in oxygen delivery.
That makes the damage much clearer. Okay, so how do these tiny invaders get around? How do they move from one alpaca to another? Is it just like breathing the same air, or?
Not so much breathing the same air. Think more like a microscopic taxi service, as you put it earlier. A big part of that is biting insects.
Oh.
Fleas, ticks, mosquitoes. They're like these little unwitting couriers. They bite an infected animal, pick up the bacteria, fly off, bite another one, and drop off their unwanted passengers.
Usual suspects.
Pretty much. But it's not just insects. There's also what we call iatrogenic transmission.
Meaning human-caused.
Essentially, yeah. Transfer of infected blood, maybe through contaminated needles, syringes, even surgical instruments if biosecurity isn't strict.
So hygiene is critical.
Absolutely crucial in vet practice. And importantly, blood transfusions from carrier animals that haven't been tested, that's another significant risk.
Okay.
And then there's pretty strong evidence for vertical transmission.
Mother to baby.
Yes, in utero. From an infected dam to her fetus. We've seen cases like Criteria, a baby camelid born to a PCR positive dam, testing positive itself for MHL at just 4 days old. That really points towards prenatal infection. It got it before it even entered the outside world.
Wow. Okay. That's quite a range of ways it can spread.
Yeah.
So if an animal does get infected, what does it actually look like? What signs should people be watching for, especially if you said many are silent carriers?
Yeah, that's the tricky part. It's highly variable. Many, maybe even most infected animals are asymptomatic carriers. The infection just sits there, subclinical, sometimes for ages.
Right.
Clinical disease mycoplasmosis, it usually only pops up when the animal's immune system is somehow compromised.
Like stress.
Exactly. Stress is a huge trigger.
Yeah.
Transport, going to shows, giving birth, weaning. Also concurrent illness, maybe poor nutrition.
So standard stressors for livestock.
Pretty much. When signs do appear, you might see things like progressive, chronic weight loss, lethargy, depression, maybe the fever, sometimes diarrhea, even decreased fertility. Now, here's maybe the most critical insight for managing this. Anemia is a key feature, yes, but it's often one of the last signs you see.
Oh, really?
Yeah. By the time you see clear signs of anemia, the disease is usually pretty advanced, which makes treatment much tougher.
So you're already behind.
You're already behind the curve. The real challenge, and where effective management needs to focus, is either catching those subtle early signs, knowing the triggers, or using sensitive tests before you see obvious illness.
I see.
Oh, and in animals with a really heavy load of bacteria, you might even see hypoglycemia, low blood sugar, because the bacteria are basically stealing the animal's glucose.
Wow.
And younger animals, cryas, they seem more likely to get the acute, severe form of the disease.
Okay, so waiting for obvious signs, particularly anemia, means you might be too late for easy intervention. That makes early diagnosis sound critical, but maybe really hard.
Extremely hard if you're just relying on looking at the animal.
So what's the best approach? If symptoms are vague or late?
Right. Because the signs are so subtle or late, the gold standard for diagnosis isn't clinical observation. It's a lab test. The polymerase chain reaction, or PCR assay.
The DNA test.
Exactly. It looks for the bacterial DNA. It's super sensitive, very specific, and it can pick up even low-level infections in those asymptomatic carriers we talked about.
Okay.
Historically, people tried using blood smear microscopy, looking under a microscope, but the sensitivity is just, well, it's really low.
Why is that?
Think about it. You're trying to spot these tiny, kind of shapeless bacteria. They're polymorphic, meaning they change shape. They're easily confused with bits of stain or other junk on the slide.
Right.
Plus, their numbers fluctuate in the blood, and they can even detach from the red cells if the sample sits too long in an EDTA tube, which is a common anticoagulant. And another thing on a smear, NHL infection can look identical to anaplasmosis, another blood parasite.
So you could misdiagnose it completely.
Easily, which really makes PCR essential for a definitive diagnosis. The takeaway, don't just trust your eyes on a slide, trust the DNA.
That makes a lot of sense. Okay, so you get a PCR diagnosis. What then? Treatment, management. Can you actually cure them? Or is it, like you said, they stay carriers? Sounds like a sticky problem.
It absolutely is a persistent problem. And here's another sort of crucial point. For animals that are clinically sick, treatment usually involves tetracycline class antibiotics. Oxytetracycline is common.
Okay.
And these antibiotics, they do help. They reduce the bacterial load. They can alleviate the clinical signs like the anemia. But, and this is a really important but, for anyone managing alpacas or llamas, treatment does not reliably clear the infection.
Ah. So they get better, but they're not cured.
Exactly. Many, probably most, treated animals remain chronic subclinical carriers.
Which means they can still spread it.
Yes. They still pose a risk of transmission to others, and crucially, they themselves are susceptible to future relapses if they get stressed again.
So it's a lifelong management thing.
Pretty much. The strategy really shifts to prevention and control. Minimize stress, easier said than done sometimes. Implement really good arthropod vector control.
Yeah.
Be super strict with biosecurity, especially avoiding needle reuse between animals.
Right.
And screening blood donors using PCR before any transfusion. That's a critical preventative step. You're basically managing a chronic condition, not eradicating it.
That's a tough reality for owners and vets. Okay, so we have this clear picture for camelids, MTEL, clinical challenge, chronic carriers. But now let's shift gears. Let's step out of the pasture and into the woods. Deer, cervids. Is it the same story, same antell causing problems, or are we looking at something completely different?
That's a great question to ask because, yeah, you're right on. It's largely a whole new cast of characters over there.
Really? No MCHEL in deer.
Nope. Studies in deer populations, cervids, they just haven't found Candidatus mycoplasma hemolome at all. Instead, deer host this surprisingly diverse range of other hemoplasma species.
Like what?
Well, for instance, Mycoplasma ovis. That's the one typically found in sheep and goats. But it's actually detected widely in lots of deer species. Captive reindeer, farmed white-tailed deer, free-ranging deer across North and South America. It seems to jump hosts quite readily.
Interesting. What else?
Then you've got species like Candidatus mycoplasma hemocervae and Candidatus M. erythrocervae. Those two were first identified in sika deer over in Japan. Distinct species.
Okay.
And it doesn't stop there. More recently, molecular surveys using PCR again on white-tailed deer in the U.S. found at least 3 different hemoplasma species infecting them at the same time.
Wow, co-infections.
Yeah, and 2 of those 3 species were previously completely undescribed.
So we're still discovering them.
Absolutely. The full diversity of these pathogens in wildlife is very much an ongoing puzzle. It's a fascinating area of discovery.
That is a lot more complex than the camelid situation. So, with all these different types floating around in wild populations, how common are these infections? Are deer getting sick like the camelids can, or is it mostly that subclinical hidden infection thing again?
Well, prevalence can be incredibly high in many deer populations. Really surprisingly high.
Like how high?
Okay, get this. One study in North Carolina found 89% prevalence in white-tailed deer.
89%.
Yeah, huge. In Japan, it was about 45% in sika deer. Studies in Brazil found rates up to 40% in free-ranging deer and even higher, 87% in some captive deer populations there.
So it's everywhere.
It's extremely common. But, and here's that key similarity back to the camelids, infections in deer are also very often subclinical.
Ah, okay, so lots of infection, but not necessarily lots of sick deer.
Pretty much. When you do see clinical disease, it's usually linked to anemia and just general ill thrift, poor condition, wasting. And again, it's more likely in animals that are young, stressed, or maybe already battling other infections or heavy parasite loads.
The vulnerable ones.
Exactly. So while the infection rate is sky high, obvious sickness isn't always the norm. It really highlights the hidden nature of these pathogens in wild ecosystems.
Okay, so if they're often subclinical and they're, well, wild animals, how on earth do you diagnose these infections? And what about management? You can't exactly round up all the deer for treatment.
You're absolutely right. It's a totally different ballgame. Diagnosis in wildlife relies almost exclusively on molecular tools like PCR.
For research, mostly.
Primarily for research and surveillance, yes. Not for diagnosing individual sick deer like you would in a vet clinic. It's about understanding population prevalence, finding out which species are there, tracking how they spread geographically.
Right.
Trying to use blood smears, like with camelids, is generally seen as unreliable for wildlife screening. Low sensitivity, plus just the practical challenges of getting good samples in the field.
Makes sense.
And as for management, here's a really stark difference. There are basically no established protocols for treating or managing hemoplasma infections in wild deer populations.
None at all.
No. The focus for wildlife health agencies isn't treating individual animals. That's usually impractical and wouldn't make a dent on a population level anyway. The focus is all on surveillance, understanding the disease ecology, how it fits into the bigger picture of that ecosystem's health.
Okay, so we've really contrasted these two worlds, the targeted clinical issue in domestic camelids and this widespread, often hidden ecological factor in wild deer. You had to boil it down. What are the absolute biggest differences that stand out to you? What should listeners really grasp from this comparison?
Okay, yeah, connecting the dots, I think several things really stand out. First, the primary pathogen itself.
Alls versus a whole bunch of others.
Exactly. All seems specific to camelids, while deer have this diverse mix. M. ovis, Saint Therese, M. hemos, sort of these novel ones. That's not just a name difference. It likely means different biology, maybe different virulence, different host interactions.
Right.
Then the host group itself is fundamental. Domesticated livestock versus wildlife. That dictates everything else.
Oh, so?
Well, it leads to completely different clinical contexts. In camelids, the focus is laser sharp on individual animal health, diagnosis treatment because these are valuable domestic animals.
Makes sense.
In deer, it shifts entirely to population health. Surveillance, ecological research. How does this impact the herd, the ecosystem?
Got it.
And while both groups often have subclinical infections, when they do get sick, the picture differs slightly. Camelid signs are pretty well documented, the anemia, weight loss, lethargy, especially under stress. In deer, it's often described more broadly as anemia and ill thrift, and frequently tangled up with other health issues, comorbidities.
Harder to pin down cause and effect, maybe.
A bit, yeah.
Hmm.
Then the diagnostic standard. PCR is key for clinical diagnosis in camelids. In deer, PCR is mainly a research and surveillance tool.
Okay.
And finally, management approach pulls apart. Active treatment with oxytetracycline plus prevention through biosecurity and vector control in camelids. Versus, well, basically no specific treatment or management protocols for wild deer populations.
Those distinctions really paint a picture of different priorities and challenges.
Okay, but here's where it gets, I think, really interesting for a lot of people listening, maybe a bit concerning. We've talked animals, animals, animals. Can these things jump to humans? What are the zoonotic implications?
Yeah, that's the natural next question, isn't it? And it does raise important public health considerations. It's a growing area of interest, definitely.
So can they?
Well, Mycoplasma ovis, the one we said is common in sheep, but also widespread in deer, that one is considered a zoonotic pathogen.
Okay. How do we know?
We have molecular evidence finding its DNA in humans, particularly in people who had a lot of close contact with animals, like farmers, veterinarians. Now, the specific risk from E. coli and camelids, that's less clear, less defined right now. But hemoplasmas as a group are increasingly being considered emerging zoonotic pathogens.
So the potential is there across the board. The potential seems to be there. It really underscores how tightly linked animal health and human health can be. Something seemingly hidden away in a wildlife population could potentially spill over and become a human health issue down the line.
That's a huge takeaway. Definitely food for thought. So, given everything we've discussed and how much we are learning, what are the biggest remaining mysteries? Where does the research need to go next? What are the big knowledge gaps?
Oh, there's still quite a few significant gaps. For one, for most hemoplasma species, including many of the ones in deer, we haven't definitively identified the specific arthropod vectors responsible for spreading them naturally.
We know insects are involved, but not which specific ones for each bacteria.
Exactly. Which tick, which flea, which fly. That's a huge missing piece of the puzzle if you want to understand and maybe control transmission.
Right. What else?
Also, the full impact. The true pathogenic effect of these infections on the overall health and dynamics of wild deer populations isn't really well understood, especially those co-infections. What happens when a deer has 2 or 3 different hemoplasmas at once? We don't really know. And then, switching back to camelids for a second, a really key practical challenge remains: developing treatments that can actually completely clear the email shell infection. Getting rid of that chronic carrier state is still a major hurdle for truly effective long-term management.
So, lots still to figure out. We've taken quite the deep dive today into hemoplasmosis, seeing how it plays out as this targeted clinical concern in domestic camelids.
Right, very focused on the individual animal.
Versus this much broader, diverse, ecologically complex phenomenon in wild deer populations.
Yeah, and that comparison really drives home the importance of a One Health perspective, doesn't it?
Absolutely.
It just highlights so clearly how the health of our domestic animals, wildlife, and us humans, it's all deeply interconnected. A hidden bug in a deer might have ripples we don't expect.
And that leaves us and you with a final provocative thought to chew on. Given how incredibly common these hemoplasmas seem to be in wildlife and this emerging zoonotic potential, how might our growing understanding of wildlife health start to directly impact human health in ways we're maybe only just beginning to grasp, especially when we know so little still about exactly how they jump around? What new questions does this deep dive spark for you about all those hidden connections out there in the natural world? Thanks for joining us on the Deep Dive.